Hello, and thanks all for joining us today in our monthly Teatime webinar. Today, we have with us Dr. Chantal Pichon. Dr. Chantal Pichon is a professor of molecular and cellular biology in University of Orléans in France, and she also the head of cell biology, molecular targets, and innovative therapies. Her main group activity is focused on nucleic acid delivery in non-viral systems. Today, she will be discussing the histidine-lipid polymers for effective in vivo and in vitro RNA therapies. So, in case you guys haven't met us, myself, I'm Suhas Wayan, and I'm the scientific specialist in EMEA's region. I also work with my colleague, AJ Johnny, Regional Sales Director, and Richard Broadhead and Ed Graham, both are field application scientists, and all of us are based in the UK. We also have our colleagues, Jürgen Schmidt, who is the Regional Sales Manager, and Martin Rabel, who is the field application scientist. Both of them are based in Germany. We also have with us Sarah Gorkin, who is the field application scientist in France. She will be taking over the Q&A session today. Over to you, Chantal.
Good morning and good afternoon to everyone. Before I start, I would like to really thank P&I for asking me to present our data today. And it's really an honor for me to start this French tour of Teatime webinar on messenger RNA on the use of precision nanosystem on microfluidics, actually. So as Sarah said, as I think it was not Sarah, it was Dinah, right? I'm really sorry. I am working in Orléans and in this center, it is called the Molecular Biophysics, and I'm leading a lab where we really work on non-viral gene delivery, and we are working on development of a delivery system, but as well as, but also on different types of nucleic acid. So today I'm going to talk about why we are producing these histidinylated polymers and lipids, and many people actually in the world is also doing this, and some of them are actually in the audience. So my first slide is just to tell you why adding histidine moiety on the delivery system. So I think you know that the histidine is one of the essential amino acids, but it has a diverse biochemical properties. And you have the first I put was pH buffering, hydrogen bonding, aromatic coordination. So, wow, what happens here? I'm so sorry. Aromaticity. There are a lot of features. And you can also see that histidine is involved in different biological systems like enzyme activity, transcriptional factors, and antimicrobial activity. So in my lab, the pioneer which has introduced this use of this histidine on a delivery system was Patrick Midou. He's here. I'm sorry, I didn't put his name, apologize. So in 1999, he started to develop histidine on a nucleic acid delivery, and he then patented it. So here in this slide, I'm just showing you that if you have a particle which is taken up by the cells, it ends up usually inside the endosomes, and from this endosome, it must to escape. And this, in this step, actually, if you have a histidine bearing particles, the histidine on the particle will be protonated because it acts as a weak basis. So usually the early endosome has a pH of 6, which is a mildly acidic. So what happens? You will have a active transportations of protons thanks to proton pump ATPases, as shown here. Maybe I have to put this pointer here. And at the same time, you also have a passive transportations of chloride. This is actually inducing what is called the proton sponge effect and osmotic swelling of the endosomes, releasing the particles inside the cytosol. But there is also another possibility. It's a fusion of the particles bearing the histidine or the imidazole moiety on the membrane of the endosomes. And as a consequence, you will have also a release of the particles inside the cytosol. So in my lab, we actually designed the different types of histidinylated delivery systems. So the first one is, of course, the histidinylated polylysine. Then we made also a histidinylated PEI, where we put different types of different moieties, residues of histidine on the backbone of a PEI. So we patented it as well, and it has been licensed by Polyteragen, which is actually the company that has been created in 2011, and Patrick is now the CEO of this company. Then we also made a lipids, lipophosphoramidate imidazolium or lipophosphoramide histamine. As you see here, there are imidazole on those lipids, and they are really acting, exactly like the particles I showed you in my previous slide. And for the messenger RNA vaccinations, we also made a mannose-elated liposomes. And you can see already for what it's for targeting dendritic cells. So these are the different types of nucleic acid formulation that we've made in my lab, as in any different types of a lab working on non-viral delivery systems. We have, of course, used the polyplexes, which is a complex between the DNA or RNA, which could be small or large, or large size, with cationic polymer. And usually we have something which end up between 50 and 150 nanometer. Or you can also use cationic liposomes. Again, we will form what is called the lipoplexes. It's a little bit bigger than the polyplexes. And for the vaccination, actually, we propose to use a lipopolyplexes, which is a kind of hybrid between a polymer and the liposomes. So first, you have to condense the DNA or the RNA with a cationic polymer to have a polyplexes, and then you add our liposomes, which encapsulate it. So here you have a cryo-tem of those particles on the liposomes here, and you end up with this lipopolyplexes. It looks like that. Or in some papers, people can call it, LPD, LPR, LPR, or LPP, but those are actually exactly the same particles composed of polyplexes and liposome. And of course, these last, I would say, few months, we started to do a LNP because we hopefully, and I'm very lucky to get the nano assembler from Precision Nanosystem. Okay, so you'll see what we have done, what we have done on that. So just briefly, before going to the messenger RNA, let me just show you why, when you had histidine on the polymers or on the liposomes, what type of features you will, those particles will acquire. So here, most of you know, it's a gel retardation or shift assay, just to allow you to see if actually your polymer or your complexes are really formed or nucleic acid is compact. So here we have different amount of the polymer. In this case, it's a histidinylated polylysine. So you see it's around 100 nanometer. And you can see that as you increase the amount of the polymer, you have the retardations of the bonds. which is bonds composing of a DNA, the relaxed and the super cold one. And here you have a complex, which is the polyplexes, which is here in this case positively charged. Okay. And the first cells that we have used for this transfection of this histidinylated polylysine was hepatocarcinoma cell type G2. It was very, very difficult to transfect. And you see here the polylysine is does not really work when you are using it to deliver plasmid DNA coding for luciferase. But if you actually put histidine of this polylysine, you increase by, from 100 to 10 to the 7 light unit, which is very high. So I told you that the endosomal escape is due to the fact that you have this proton sponge and the TPS is really working on is involved in this proton sponge effect. So if you inhibit the endosomal acidification by using bafilomycin, which is an antibiotics, the proton sponge, the ATPase, proton ATPase, V ATPase doesn't work anymore, so we won't have this proton sponge effect. So you can see here that from the 10 to the 7 light unit, you really disrupt actually the gene transfer efficiency. Below, I'm just going to show you what we can also have if you if you modified the LPI with histidine. You see here that you are increasing the buffering capacity. So here it's a titration experiment where you have your pH and here a different amount of your sodium hydroxide. So clearly you see the improvement of a buffering capacity thanks to the substitutions of a PI with histidine. So what is really interesting is that when you do this, you increase the expression of gene delivery in a different, the expression of the efficiency of your gene transfer in the different cell lines, as you see here. HeLa cells, heck, even the HEC293, the C2C12 doesn't really work, and also for the cystic fibrosis epithelial cells. But I think what is really interesting is the fact that when you substitute your LPI with histidine, you highly reduce actually the mortality or cytotoxicity induced by the PI. You can see it very, very clearly, and especially for cells that are very fragile, like C2C12, which is a urine myoblast, but also the same thing can occur with a CFTE. This one is actually the cell line that you use that have a mutations of CFTR for cystic fibrosis. This table just shows you that you have a very nice effect on the substitutions of histidine on the PI because you have a higher, you can use higher amount of your PI without toxicity, as you can see here. So this PI, histidinylated PI, has been, as I told you, licensed by Polyteragen, and actually they are using it for viral vectors production. I'm going to switch on the LPR formulation for anti-tumor vaccination. We actually published different types of papers on that, and it started with the first one in 2007, where we showed that we can inject systematically, I mean, by intravenous injection. The formulation made with lipoplexes, coding for MART1 mRNA, without any targeting, made with those lipids, liposomes. And then in 2011, with Federico Perso, the first one has been done with a PhD thesis of Michael Mouquet. Then in 2011, we showed that if we add mannose-related motif to get a targeting of dendritic cells, we can improve the vaccination obtained with this LPR. And the last one was the adding tri-mannose-related lipids. And this has been performed in collaborations with the Tillmans lab in Brussels with Karin Brekpot. And here is Kevin van der Riot, who has done his PhD thesis at that time. And it's also with Etherna. So it was really a very nice story from, starting from now targeting and improving it up to the tri-mannose-related motif. I'm going to show you what are actually the data. So as I said, we decided to use these lipoplexes because you see the cationic polymer with a messenger RNA hardly work in our hands. So by doing this, we found that indeed we are able to compact the messenger RNA, to stabilize it and to protect it, as shown here with this electrophoresis, agarose-gel electrophoresis. You see the messenger RNA here, LPR, the RNAs, and clearly you protect it. And if you extract the messenger RNA from the formulations, you do have a very nice messenger RNA which is intact. Here is the physicochemical characterizations of the particles. It's around 150 with or without mannose-related motif on it, and the zeta potentials are actually 25, 24 millivolt. Okay, so we can have a difference between the size according to the type of messenger RNA that we are using. In this case, it was EGFP. For MART1, it's a little bit lower, smaller, both for the size and the zeta potentials is also less cationic. So the first model that we have used was B16F10 melanoma model, and we chose to use a MART1 melan A because it's shared with 90% of a patient and it has 68% of homology between the murine and the human one. So this is actually the structures of mRNA that we used. We use here, ANARCA, which is a CAP0 analog, MART1, and 100 poly A at the 3'. Okay. So this is the protocols that you use. Day 0, I would like to stress out, this is an IV injection. The vaccination. Day 7, we make the boost, and day 14, we did the challenge. Okay. So the first study shows that we actually were able to prime specific CD4 and CD8 T cells, as you see here. In the lymph node, 3.5, 3.8% of CD4 or CD8 plus positive cells, respectively. Also in a splenocyte. And if you assess the cell immune response in terms of lysis, you see here that the messenger, when you use a messenger RNA coding for the MART1, we have a very nice lysis of the cancer cells compared to the irrelevant messenger RNA and also the buffer. In terms of tumor volumes, we delayed very significantly the growth of the tumors when we use the injections of 12.5 microgram messenger RNA per mice using our formulation. So this one is the marked one, this one is irrelevant, which is also the luciferase. Interestingly, we find out that the only formulation that is really working was actually this histidinylated LPR, nor the lipoplexes or the polyplexes didn't really work for that. So then because we need to reach dendritic cells, we decided to make the targeting. So the first targeting, which was the most easiest one, was to use just a monomethylated motif on our liposomes. So we did exactly the same protocol. And when you harvest the spleen cells and look how many positive dendritic cells have been transfected with a messenger RNA, so here each messenger RNA coding for EGFP, you can see that we have improved a lot the amount of dendritic cells. So from 2.5, we reached 13% in terms of positive cells. So what is the outcome of that? So in terms of volume growth, we see that again, we have a delay of the growth, even though it started to grow at 23 days, unfortunately. But clearly, you see here again, the survival was really good because, it's from 26 days where you end up more than 30 days here. So from that, we think that maybe we should do another improve the targeting. And if you look at the literature, actually the ligands for the mannose receptor that you would like to reach on the dendritic cells, it's much more complicated than the monomannose motif. Here it is. It has more than 5 mannose, 6 mannose, 7 mannose. And so we decided to produce a lipids variant for 3 mannoses, which is not so easy, but we managed to do that. Okay. So again, we did exactly the same protocol, and this is actually the work we done with Chris Tillman's lab, and Kevin van der Woert was the first author of this. So you can see here, again, if you do the injection, all of the formulation ends up in a spleen, and it's expressed in the spleen. No expression in the liver, which is really very nice because you have an exclusive delivery in the spleen, reducing a lot of different types of inflammation and cytotoxicity. I'm sorry I didn't tell you here. Again, in terms of physicochemical features, we do have 200 nanometer, and it's very positively charged, of course. To show that the dendritic cells are really the cells that are transfected here, we use the transgenic mice expressing CD11C fused with DTR, which is actually the receptor of the tetany toxin, of diphtheria toxin. And you see that if you eradicate the dendritic cells by adding the diphtheria toxin, it's really, there is no more signaling. This systemic LPR administration instigates a potent T-cell immunity, as shown here. Here it's a delivery of OVA messenger RNA, when you take the blood of the mice, you see that as soon as 15 days, you induce a OVA-specific CD8 cells, okay? And if you do a recall after 60 days, you also have an increase, again, the productions of these OVA-specific CD8 T cells, indicating that you do have actually instigates a memory. The same thing is occurring if you take the spleen, harvest it, and see. You can really re-stimulate both cells using the peptide over peptide. You see the increase of interferon gamma and not very much of TNF alpha, which is very good. And here is the two stimulation. So the nanostring experiment shows that we do have an upregulation of interleukin 12, which is the most polarizing cytokine, driving Th1 and cytolytic T cell response, very marginal increase of inflammatory cells, and no induction of Th2 and Th17 oriented cytokine, and of course a very good Th1 oriented cytokine. What is interesting actually with those lipopolyplexes is the fact that you can use both modified and non-modified nucleotide. And here it's I'm just going to show you an experiment which is a therapeutic vaccination, because here we implant the cells, the cancer cells, and then you treat. Always a free treatment, spaced with 5 days. We use here lipoplexes or lipopolyplexes. So if you look at the E7, because here we express E7 messenger RNA related to the cancer cells. So you can see here that only the LPR, both unmodified and modified with N1 methyl pseudouridine, actually induce the productions of E7 specific CD8 T cells. Concerning the tumor growth, very clearly you see here that the mice that has been treated with the LPR coding for the E7 messenger RNA with the without actually the mRNA, with modified mRNA or not, there is no increase, there is no growth of the tumors, which is not the case again for lipoplexes. And I think it's good to point out that if you use lipoplexes, if you use a modified messenger RNA, it didn't really work. You know, this one, the brown one compared to the green one. With another model, which is a B16 ova expressing cells, we exactly have the same trend, even though actually in terms of response, it's a little bit lower compared to TC1. I guess because this one is very aggressive. The hematocompatibility of those tri-man LPR has been assessed in the human blood, and we have seen that actually there is no C3A cleavage, which is the, one cleavage of a component of complement here, so which is really interesting. You can see here, compared to the CVF, the venom from factor. Here is a quantification up to 223 nanogram per ml. There is no also hemolysis determined through the measurement of hemoglobin release on lithium heparin anticoagulated blood. As you see here, this is hemolytic threshold, and we are really, really below. And it's the same if you look the color again compared to the collagen. So I think we have a very safe, I would say, particles. So this first summary shows that we do have a very nice particles that are able to stabilize and to protect from RNAs. They can be used IV. And I didn't show you, but they are working even in intradermal or intramuscular injection. The second part, and I will be a little bit quicker because it looks like I'm very late. We also applied those LPR for to accelerate bone regeneration. So There are a large demand on that because if you have a larger non-human fracture, it's very hard to self-heal, and you require bone tissue engineering. So you can use autograft or allograft, but autograft is a little bit difficult because there are different types of limitation. So this is a work from Pin Wang and Federico Persch, who I've seen him before, in collaborations with B3OA and Delphine Rogart in Paris. So what we've done, instead of using chemically modified messenger RNA to avoid the immune sensor activation and increase the translation, we decided to use NS1 mRNA. NS1 is actually the protein that is used by influenza virus to inhibit all of the innate immune sensor during its infection. So you will see that when you co-deliver messenger RNA of interest, like here in this case BMP2 and NS1 messenger RNA, the NS1 can inhibit all of the innate sensor here, and actually you have exactly the same effect as when we are using modified nucleoside. So we put the two messenger RNA inside the porous matrix and use it to induce ectopic bone formation. So first of all, we tried to understand and we tried to make optimize the amount of BMP2 mRNA and NS1. And you see here at 0.75 plus of BMP2 mRNA plus 0.25 microgram NS1 mRNA was the optimal, and we able to secrete BMP2 as you can see here in the supernatant of mesenchymal cells. At 14 days, we have a alkaline phosphatase activity, and also an extracellular calcium staining actually here at 28 days, indicating that it's working. We implant this, I have a problem. We implant this in our scaffold, and once it's in the scaffold, we have found that indeed there is a vascularization that have occurred, clearly seen here. And I think it's by micro-CT, we show that there is an increase of productions of bone, which is an ectopic bone. Okay. So today, Federico Persch had recently a grant, and his goal is really to formulate those messenger RNA, find out different types of compositions of transcription factor for bone regeneration, and this will be done in collaborations with RMS Nantes and also B3OA in Paris. For my last part, I would like to present you what we have done for with the microfluidic system, because even if it's working in the different studies that I have shown you, we actually found out, as in different groups, that sometimes there is an heterogeneity of our formulation. So first of all, we tried to really make optimizing the formulation of lipoplexes and use those formulated lipoplexes in the microfluidics to do our lipopolyplexes. So here, just showing you that we have stabilized up to four months. I think it's much more than that now. Very low dispersity, and you see very nicely, and exactly the same in terms of potential, zeta potential. If you look at the transfection of dendritic cells that is shown here, compared to lipofectamine messenger marks used as a benchmark, you see here that we have a very high improvement of the percentage of dendritic cells, which is here in orange, and you see that the mortality is really, really very low. And this is different types of lipopolyplexes. If you take those lipoplexes or liposomes and made the lipopolyplexes with those, we see here again, and very interestingly, here this one is the one made classically with film hydration. you see that again, we have a very high improvement. And again, this one is not optimized one. So we are really very happy because we find out 2 leads that can be really used for our next experiment. We decided to open the different types of cell lines or cells used for those messenger RNA with those different types of formulation. And here, it's a work made by Nabila Laroui, which is a postdoc in my lab, and Christophe Delehet, which is a PhD student, in Schwann cells, keratinocyte and fibroblasts. And again, you can see here that we do have a very high expressions of messenger RNA here with high MFI, which is related to the copies of NGFP. Again, here is a lipofectamine, and it is working even at 0.25. Okay. Schwann cells, LPR, it's a little bit difficult, but we found out this ratio, which allows us to have a 93% and very high MFI. Keratinocyte lipopolyplex is the best compared to LPR, and the fibroblast, the lipopolyplex is better, much better, I would say, than the LPR. Still, the LPR is not so bad. So now, if you would like to use it for skin cells, we need to find out the optimized combination between all of those different types of formulation. Then we switch to the productions of LNP. So here are different types of LNP. I'm so sorry, cannot disclose it, of course. And you see we have a neutral, almost a neutral zeta potential, very high encapsulation. And you see, again, we have very nice amount of size of those LNP. Some of them doesn't work, but you know, it's an optimization. Those LNP actually can really can also be very efficient in natural killer cells. So I'm going just to show you data that we obtained with KIG1 cells. So here, the first one is just to make a kind of benchmark in lipofectamine, screenfect, and or CD6.1. And you see here very clearly that the CD6.1 has a very 60, almost 80% of transcription efficiency, very high MFE. And I think what I really like is the very low mortality. This is for 4 hours. And here, if you look, this one is another one. This one is a lipoplex. This one is an LNP. You see here that even after overnight, we have very low mortality and high transcription efficiency. So we are very excited with those data, and now we are using it for primary cells, and our application are ongoing for cell therapy, of course. So the second summary is just to tell you that I think we managed to, instead of using modified nucleotide, we managed to use NS1 mRNA to inhibit innate sensors, combined with messenger RNA of interest. Or LPR can be loaded into collagen scaffold. I did not have time to show all of this, but I am really ready to respond to you, answer your questions. The RAM containing BMP2 and NS1 mRNA can induce de novo ectopic bone formation, and now you would like to heal fractures with those formulation. And I think now every formulation that we are going to make in our lab will be done with microfluidic system, because it's really improved the formulation, both for lipopolyplexes, but also the LNP. And the stability is, the data I have now is for 4 months, but it's still ongoing. And we are now doing a lyophilization process as well. So very interesting to see the LNP, which actually have very efficient for dendritic cell, NK cells, Schwann cells, and skin cells. And again, we have an in vivo application is made by Nabil Alaoui, going for skin diseases, and Christophe and Ivan Chigenek will do the cell-based therapy. I think that was my last slide, but before I finished, I would like to thank, of course, my team, great team, very involved, very hard worker, Patrick Midou, Federico Perche, Anthony Delalande. I didn't talk to what she's doing, but he's doing a ultrasound delivery system. All of our support, because we cannot do anything without them. These are our collaborations, and a special thanks to people from Sanofi, Luc Even and Nathalie Ramex. And I would like to thank you for your attention, and I'm so sorry for being late. Thank you.
As we are over time, I think we need to finish it. I would like to thank again to Professor Chantal Pichon, and I would like to thank again for your participation. Our Teatime webinar is a monthly event, so please keep an eye for the next one. Merci beaucoup pour votre participation. See you soon. À très bientôt.
Merci beaucoup. Thank you. Bye-bye.